Today's lesson is looking at lipids and the structure of which lipids there that exist in nature. Again, lipids are primarily the energy storage, long-term energy storage for most organisms. They also form protective barriers such as waxes on the outsides of leaves, and phospholipids, which are in cell membranes. Those are all typically fatty acid based fats, or lipids I should say. And there are hormones and other chemical indicators that are steroids. Cholesterol is an example of one. Those are also lipids. We use lipids also for insulation, and fat-soluble vitamins are found within it. And just a reminder that your fat-soluble vitamins are A, D, E, and K. That's more trivia. You won't be tested on that in my class anyway. And steroids, those are ringed structures, ringed carbons. So the only real thing that keeps all lipids together when you think about it is that they're primarily nonpolar. Okay, being nonpolar means pretty much one thing: they don't like polar. So when you're looking at things such as water and oil mixing together, they don't mix together because polar and nonpolar do not mix. So again, fats and oils we'll discuss, particularly in structures. Fats quite often found in animals, oils found more in plants. We have waxes, those are nonpolar entities. Phospholipids, they're the sort of odd one in the bunch because phospholipids have a polar end and nonpolar ends. And then steroids, again, those are your ringed structures. So let's quickly take at triglycerides, which are fats and oils. And when you're looking at triglycerides, you can't really call them a polymer. Like when we talked about starch versus glucose, starch being the polysaccharide and glucose being the monosaccharide, and so a polymer is a whole bunch of glucose molecules stuck together. In oils and fats, the triglyceride is basically built with a glycerol, and you're going to add, through dehydration synthesis or condensation, 3 fatty acids. Okay? And so when we do that, when you're adding the glycerol and 3 fatty acids, you have a glycerol. Glycerol is 3 carbon alcohol, and each carbon has a hydroxyl group attached to it. Okay, so you've got 3 carbons, 3 hydroxyl groups, and then you're going to add a fatty acid to each one of those. And a way to remember a fatty acid, whenever you think of acid in biology, not whenever, that's a hyperbole, but a lot of times when we're talking molecular structure, an acid is a carboxylic acid group. So remember, you've got a carboxylic acid, which is a carbon, a double bonded oxygen, and a hydroxyl or an OH attached to it. That's the acid. And then the fatty side of things is the carbon-hydrogen linked chain that looks like this. And fatty, when you think about it, carbon-hydrogen bond sites, they store a lot of energy. So that's where you can think of the fatty part of it is the hydrocarbon, and the acid is the carboxyl group. And if you did that 3 times, because you need to bond them, I'm just going to draw a whole bunch of carbons here. I'm not going to add the hydrogens. And then again, I'm going to take this. I'll put, rushing through. I need to stop rushing. I'll put it just down below. And then you have your carbons, And now you see your glycerol, which I'm going to put in blue underneath, and then your fatty acid, which is the green on this side. And when you join them together, again, remember when you're joining, you're going to take that water molecule out, so you'll end up making plus 3 waters, right? And that bond is going to change, so I'm just going to draw one of them here. So the glycerol would keep going down this way, okay? And then you would get the O, and then the carbon with the double bond O, and C, 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 C. And then if you kept going, you would get another I'll put it up here. I might as well draw it out because then it's sort of clear to see. C, 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 C. So as you can see, you have a new formed thing with the 3. Do not forget you have formed 3 water molecules. Now, the bottom here it says waxes are synthesized by long chain fatty acid and a primary alcohol. Don't worry about that. We're not going to ask you to form that in this class. So I'm just going to demonstrate the difference between a fat versus the oil. And things when we look at that, fats typically are animal-based, not always, but okay, and oils are typically plant-based. Fats are quite often solid at room temperature, and oils, not always, but quite often are liquid or can be presented liquid fairly quickly. And fats, because of being solid in animal, are often known as saturated fats. And oils are quite often unsaturated. And the concept of saturated versus unsaturated is nothing more than looking at a fatty acid chain. Okay? When you look at the fatty acid, normally what you have is a whole bunch of carbons that stretch across. Okay? And if I were to add all of these dashes showing representation of hydrogens, this fatty acid chain would have a whole bunch of hydrogens. And in fact, it would be full of hydrogens. Well, if it's full of hydrogens, then another word of full is saturated. Okay? So when you look at a fatty acid chain and it's all single carbon-to-carbon bonds, that means it can hold no more hydrogens, which means it's full. So if there are no carbon-to-carbon double bonds and it's all single, we call that a saturated fatty acid. Okay? And if you take it and then you look at it and go, oh, wait a minute, I'm going to add a double bond here. Okay, so if you see I've put a double bond blue in there, by doing that, I have to be able to remove 2 spots for hydrogens. Now hopefully you saw that I removed one here and I removed one here. I arbitrarily moved one on either side. It's not done that way. And when you do that, it now means there is possible space for more hydrogens. Not that there is, but there conceivably has more space because you've now added that double bond there. So you've lost 2 hydrogens. That makes that fatty acid unsaturated. Okay? And by unsaturating it, all you've done is you've changed the properties of that fatty acid. And if I was to draw the saturated fatty acid out, just as a chain, you would have pretty much a straight line. Okay? If I were to draw out the unsaturated, as soon as where the double bond forms, you have a bond here, and then that double bond causes it to move and it changes shape. And so you actually get a kink going in there. And an interesting point is when you have unsaturated fatty acids, those kinks cause the triglyceride, because they're all, both fats and oils are triglycerides, to actually get larger in volume. That makes it less dense, and that less dense actually makes it more chance to be a liquid. And that's one of the reasons why many oils are actually liquids and not solids, because the space has moved out. Okay, hopefully that makes a bit more sense in terms of fats and oils. And remember, the fats and oils, they are both triglycerides. Okay, so we've talked all about triglycerides here. Now, let's move on, and we're going to talk about a specialized, it's not a triglyceride, it's more of a diglyceride, and those are the phospholipids. Because phospholipids are hugely important to life as we know it, because most cell membranes are actually a phospholipid bilayer. And so we're going to look at the structure of the phospholipids. I'm going to talk a little bit about steroids first, and then we'll come back to the phospholipids. The steroids, they're ringed structures, okay? Cholesterol is again, as I said, cholesterol was the most abundant of steroids, and we'll come back to steroids again, especially in homeostasis. You won't have to draw them. You should be able to recognize them. So if you're taking a look for the 4 rings of carbons. Take a look in references, and you will be able to see that. Okay, let's flip to a phospholipid just as an example. Okay, so phospholipid, if we're going to sketch our phospholipid, and if you look at those cell membrane diagrams, especially when we get going into cell membranes shortly, you will see that they're quite often drawn like this. And if I zoom into one of those, and I do something like this, well, the head is hydrophilic. Okay? And the 2 tails are hydrophobic. And if you were to look at that, the head is nothing more than the glycerol. Oops, I'm going to color code that properly. Okay, so let's go back to this and we'll go black. So remember it's glycerol, and then the 2 tails that are hydrophobic are actually a fatty acid. I'm drawing sideways, I hope that's okay. And if you think about it, that's just showing the 2 tails being hydrophobic. On the end here, I'm just going to flip it over and take it on the top. The O, if I put it up here, is attached to a phosphate. And if you remember, a phosphate has 5 bond sites, and being negative, it makes this end polar, and the 2 tails that stick down as nonpolar. And so your phospholipid is nothing more than your glycerol molecule, which is right here, your 2 fatty acid chains that run down from the 2 carbons, those are your nonpolars, or the hydrophobic ends, and the head, which is a phosphate group, a phosphate-containing group, which makes it polar, and that's your hydrophilic end. And that basically ends lipids as a quick crash course on structures. Hopefully that was good, and don't forget, keep smiling.